So I'm just going to talk a little bit about preprocessing and denoising of Neuropixels data. I should have said I'm Jennifer Colonell. I work in Tim Harris's lab at the Genelia Research Campus. It sort of goes without saying, but I want to say it anyway, that the best denoising technique is to fix your problem before you record. There's a standard set of tips that we always tell people to look at: soldering, look for pickup from the room, make sure that you understand how your animal is grounded, and that should generally only through the probe. And there's good information about this stuff in talks from previous UCL courses, and so I've put some links to those, and I already have these slides up on the Slack, so you can go look at those. So one of the first steps in doing your preprocessing is bandpass filtering. This is pretty basic. Of course, what you're trying to do is to remove everything from the recording except the frequency portion that you need for sorting. So you're taking out the low frequency noise like line noise and but also the LFP, and on the high frequency end, you're mostly removing noise. And in all of these slides, I'm showing you examples, mostly from this recording, which is from Susu Chen from Striatum. And so you can see that after bandpass filtering, we've got beautiful flat baselines and very beautiful spikes. It, however, doesn't get rid of these big artifacts, which are due to a lick detector. And that's because the amplitude of those is too big, so they kind of leak through the filter. So we'll need to take care of those in other ways. A couple of notes about bandpass filtering: you need to use an acausal filter so you don't get delays relative to the raw data. That means that it doesn't introduce phase shift. That's the Butterworth filters in CatGT, which is the preprocessing utility that sort of goes along with Spike GLX. Those are acausal. They're the ones implemented in Spike Interface are also acausal. The waveforms will change a little bit, and how much they change is a function of the limits that you put on your bandpass filter. The broader the frequency range you keep, the more they'll look like the native waveforms. But if you're doing very detailed waveform analysis, you might want to consider, even if you've had to filter a lot to get rid of noise before you did your spike sorting, you might want to go back to the raw binary or the raw binary that has actually been through T-shift, which I'll discuss on the next slide, to get your waveforms. This is just an example at the bottom. The blue is the raw waveform. The dashed orange is the data that's been through a 300 to 10 kilohertz filter. Looks just like the raw data. When you go down to like 6K, you do start to clip off these high frequency regions like the sharp trough and this little pre-peak. So why would you do that? Well, you do it because you do improve the noise a little bit. Those numbers there, I've estimated the noise from the median absolute deviation for this and compared the 6 kilohertz to the 10 kilohertz filter, and you do get rid of another 1.5 microvolts of noise, which is great. You also lose a few units. So the way I'm quantifying unit loss for this and for a slide down the road is I've sorted with kilosort 4, and then I'm counting up good units using the IBL quality metrics to say who's good. So you lose a few units, but not too many with the lower limit on the bandpass. I talked about T-shift, and I wanted to provide a slide here about what that looks like. So T-shift or rephasing, what we're doing here is we're correcting for an aspect of taking data on Neuropixels probes, which is that we have that not all of the channels are sampled at exactly the same time. So the 384 channels are actually feeding 20 on a Neuropixels 2 probe, are feeding 24 analog to digital converters. And it does that by taking sets of 16 channels and sampling them at slightly different times through the 33 microsecond sample period that makes that 30 kilohertz sample rate. And the effect of that in the raw data, you can see that over here on the left, is that if you look at, and you do have to look carefully at specific channels, but if you look at channels that are sampled early, in this case 224, versus later in this set of, earlier versus later in the sample period, the peaks will look different. They don't line up. And after you do the phase shift correction, which Winter has described very well in one of his papers, those line up perfectly. So this is a really important thing to do, both to understand how your spikes are propagating across the probe, and then also if there's underlying noise, it will line that noise up better so that it gets subtracted off more effectively when you're doing spatial filtering. CatGT does that by default. Spike Interface and of course the IBL pipeline have similar functions. So moving from bandpass filtering onto spatial filtering, I'm going to in particular describe the modes we have in CatGT, but there are similar options in other tools. Spatial filtering is the main way that we denoise data, and it's because the main clue that we have that a signal is noise rather than actual neural activity is it tends to be spread over more channels. You'll see noise all the way across the probe, and real signals are confined to a smaller area nearby the unit that's firing. So the three versions in CatGT are what we call global CAR, but it's actually a global median reference. And what it does is for each time point, you take all of your 384 channels, actually 383 minus the reference, but you take all the channels and you find the median of that distribution, and you subtract it off of all the channels. And that works great when all of your noise looks the same across all the channels. Global demux is a variant of that, where we take the groups of channels that were sampled simultaneously, as I described on the previous slide, and we subtract the average of those simultaneously sampled channels from each channel in that group. And then there's a third variant called local CAR, where for each time point and for each channel, we take a center channel and we average up signals in a region that is defined by an inner and outer radius. We take all of the sites that are inside of that donut, average the signals, and subtract them off. I usually don't recommend local CAR because you are liable to subtract off signal from your neuron as well as noise. And I have a little illustration of that here. I took two recordings. Again, these are from the MAP project from Susu Chen, and sorted them after doing my noise reduction with global CAR versus local CAR. And in the striatum, it's not too bad, but in the cortex, where in this recording at least, we have a lot of neurons that have very big spatial extent. We lose a bunch of good units, and that's happening because we're actually losing a bunch of the signal from those units. And so there's two messages there. Maybe you don't want to use local CAR, but the more important message to me is when you're doing more severe filtering like that, it's a good idea to go and actually quantify what's happening in the sorting. So I'm mostly recommending global CAR and global demux. I wanted to talk about the differences between those two. In the upper right here is T-shiftless global CAR, and then this is the same thing with global demux. And you can see that this first artifact is not all the way removed by global CAR, and it is removed with global demux. The second one is removed pretty well with both. I've also quantified the change in noise, and this is looking at improvement in the noise over just doing the bandpass filter. And global demux always gives you slightly better reduction in noise over global CAR. They both do a good job reducing the noise from the original just bandpass data. So that leads to the question, one more thing. So why is that? Why is global demux working better? And the answer is, with global demux, the answer is when we have high frequency features, they're undersampled at that 30 kilohertz sample rate. And so artifacts are generally super high frequency, well, not generally. Electrical and light artifacts are often contain super high frequencies. And if we look at the waveforms of those, this is one of those licking artifacts that I showed you in the other pictures. When we look at those up close, we can see that as we look across the channels, the waveforms look really different. And the places where they look the same, like 0 and 32, is actually because those two channels are sampled simultaneously. And I have a cartoon of how that happens that I won't walk through in detail in interest of time. But basically what's happening is because the artifact is so fast, the signal is so fast, we sample different parts of it when we sample it at different times, which is what is happening in different channels. Phase shift, the phase correction can't correct this because we actually don't have all of the information. And so that's why global CAR can't fix this entirely, but global demux can. So why don't we always use global demux? Well, we don't always use global demux is because it samples fewer channels. And that means if there's a big spike on one of those, you're going to make ghost spikes other places in your data. And that's really inconvenient. We still keep it in CatGT because occasionally you have data that looks like this, where there's all kinds of terrible high frequency noise all the way through the data, and then global demux is the only way to handle it. In all other cases, global CAR is a better option after you've done the phase correction. And that's because it's using more channels and it uses the median. So when you have isolated high frequency artifacts like the ones I'm showing you, those are better handled by identifying those and removing them. So CatGT has a tool to do that too. It's called gfix. And what gfix does is it allows you to it identifies artifacts by looking for things that have big amplitude, big slope. They tend to be sharper and have high frequencies, like I said, and then it defines the extent of the artifact to the region that it's going to have to clip out by where it's going to return to baseline. And you get to set all of those parameters based on how things look in your data and where those conditions are met across 25% of the channels. CatGT identifies those, and then it will replace those with a line fit, or if you prefer, you can just put zeros in there. And this is just a picture of what that looks like. This is actually that same artifact on another channel. It's identified it, and after it's fixed it, what it has done is it's replaced that region with a straight line. So there's two limitations to this form of artifact reduction. One is your artifacts might not fit that model perfectly. If that's the case, we made another tool that will just let you hand it, it's called overstrike, and you can hand it a list of times where you need to take out artifacts. Those can be hard to identify from the initial raw data, but if they're the result of something that you are doing, like you are turning a light on and off, which is what my example is here, is we're turning a laser on and off, and as the laser turns on, it creates these big artifacts here, then you can analyze your data. CatGT can pull off these edges for you from your NI data, for example, and then you could apply that using this little overstrike tool. There is another limitation, which is we're zeroing out the artifact plus a little bit of time after it. And if there are spikes there that you need to analyze, for example, it's electrical stimulation, you're expecting spikes very soon after that, then you actually need to fit your artifacts and subtract those off. And this reference here from the Colonell lab is a really great example of that, and they have a published tool as well. So some final thoughts on it. For bandpass filtering and phase correction and spatial filtering, use the available tools. They've already handled a bunch of complications that come up when you are handling large data sets. They also read the metadata and digest it so that the phase correction, these things all depend on the hardware of the probe, and they are subtly different between different probe types. And the tools that are out there, including CatGT, handle that by reading the metadata and looking up how to do it. For noise reduction, of course, always fix your rig if you can. The standard set of parameters in CatGT are a good place to start. If you feel like your noise is interfering with your sorting, the things to consider are looking at a narrower bandpass filter, so like 300 to 10 kilohertz, or sorry, going from 300 to 10 kilohertz to 300 to 6 kilohertz. I've even seen people go down to 3 kilohertz. If you have some kind of horrendous high frequency noise problem, you can try global demux, and you know the problems that can go along with that. But I think the most important message for my talk is, however you're fiddling these parameters, you really can go in and estimate what the changes are to the sorting, and so that's what I really always recommend. And these are just references about all the stuff that I did in this talk, and again, the slides are already on the Slack, so you can go and read more details about all that stuff. And that's everything I have.